with the image of the week in a way. If you missed it on the news, it's announced yesterday the evidence of the cosmic inflation, the start of the Big Bang, which was its recording from the trillionth second after the bang happened. I thought I worked small, but that's really a long way away and very small. I'm conscious of the fact that I work within a continuum of artists and scientists working together, and in a way each have informed each other's practice and at times anticipated each other's areas of interest and ways of imaging those areas, almost predicting what the other might do at times. And I work within that continuum, and I suppose I became interested in the nature of representation and the representation of nature, you might say. But also the hierarchies and status of the images produced. There's an interesting book, catalogue of an exhibition, Molecular Aesthetics by Peter Weigel, and he suggests that between 1500 and 2000, art mainly limited itself to individual expression of representing the shapes of visible things. And whereas the same period, science concerned itself increasingly with objects and creatures that the eye could not see. I think that's a little simplistic, but I can see where it's coming from. When he goes on to describing the way that the liberal arts morphed into the fine arts, and when that happened, the fine arts stole the aesthetic moral high ground to decide what was visually valuable. And in a way, it was an unfortunate time. I think it's an interesting time. In the same way that relationship between sculpture and artisan. And I think those issues of aesthetic hierarchies are not helped by people within the different disciplines. So there's a huge catalogue of an exhibition from botanical illustrations from the Botanical Garden in Vienna, at Water Lake. And he describes botanical illustrations as having very little to do with art, but belong rather to the realm of sciences. Aesthetic considerations are wholly inappropriate, beauty is a pleasant but also wholly irrelevant side effect. Leaving aside what beauty might be, I think he's fundamentally wrong. I understand why he might take that position, and I did tell him that I've dined out on that quite a few times, but I think it's a fundamental mistake. So it's within that context that I work. This is by Pietro Mattioli, and it's from his publication Eclectic from 1568. It's a big herbarium book. It's a hand-coloured woodcut. I bought this in Parma in the summer. It's one page, the one before. And it's based on the work of Discoridis, which is 1500 years earlier. So trying to describe the plants that Discoridis was depicted. So the relationship between medicine and visualization of the objects and the plants is very important. And this is the plant Ampelophrasum. It's a wild leek. It's a fresh flower. That's a dried seed head. And those are the seeds. And that's the seed coat, that's the surface of the seed. And I know last time we talked about, it was suggested that Leonardo was maybe not the best example for interdisciplinarity. But there's a nice quote by Philip Ball, science writer, when he said that all you had to do is sit and stare for hours, not to see things more sharply as it were, but to stop seeing, to transcend the limitations of his eyes. And for me, looking is really important to what I do. And I think I realised how important when I started using microscopes, that I actually have two reasonably good lenses, supported by other lenses in front of my eyes. So what I'm trying to try and do is to describe my process. This is outside my studio in Greece, where I'm very fortunate to have space to work. So the process starts off, I'll go and collect. The studio is surrounded by wild orchids. And I'll take those back into the studio, and I draw them using the process of developed using mixing various dyes with Indian ink. And I'll just paint directly, very, very quickly, and then after a minute or 2 minutes, I'll just wash off under the tap, wash off the excess to get a drawing which captures the spirit of that plant, but also in quite an accurate way, but without it being kind of laboured. And I also use the camera, and I use it to assist looking and to record the material that I'm working from. I can't always identify it at the time. And to look more closely at who inhabits the material that I'm working with. And I use the camera to go in even closer. This is a butterfly orchid. And then I use the microscope to go in closer again. So these are trichome hairs on the surface of that petal, which in a sense are partially navigation aids for insects penetrating the kind of flower. And then I'll take sections through the stem of that flower to reveal the kind of cellular structure. And this is just another example of an anemone. And a drawing of it. And the micrograph of the anther and a pollen grain from that flower in that original picture. Very well known image of Robert Hooke from his micrograph here, which you'll be able to see here, the book, next January for an exhibition that Maria Wallace is curating, called Lens on Light, thank you. What I find interesting about this, partly not because of the way he kind of describes cells and core and monastic cells, but really that he's shown two orders of magnification, a magnified image of the cork and plant side by side. So you're seeing through two different lenses, as it were. And I use that as a kind of format for portraying images. I was really lucky, about 15 years ago I wrote into Q suggesting that they might like to work with an artist. And I got one reply from all the letters, but that changed my kind of life career, really. She was the head of research into pollen. So she invited me in, we were kind of mutual Trojan horses. And she trained me to use the scanning electron microscope. It's not that difficult. And I started developing, just gathering my material, and I would place the image and the photograph and the pollen grain side by side. And when I first started, I was working, I did all my own preparation, and I think I was, obviously, if you know anything about electron microscopy, traditionally you'd end up with from the microscope with a black and white image, to which I am introducing colour. And I was a little cagey to begin with. I didn't want to be seen to be maybe taking too many liberties, too many scientific liberties. But I realised that actually there are commonalities. Madeline describes surfaces as kind of sculptured and ornamental, but she would never show a pollen grain like this, really, because it's collapsed. And actually that's what interested me, its form. So gradually I started to manipulate the material more intensively, partly as I developed the kind of process. It's very straightforward, it's just kind of coloration through Photoshop, but using the same sensitivities and sensibilities that I might use for drawing. When I started to show these, people would start to ask the obvious question, is this the natural colour? Is this the real colour? So I would say, no. And so they say it's artificial colour. So I say, well, and I started to take exceptions to that, because what that implies is that it's a coloration through a program, scientific program, which is a fairly standard scientific description of false coloration. They are limited by the programs and by their ability to kind of use them, perhaps. I don't have those limitations. So I'm using colour in the way that the plant might use colour to attract an audience. The plant will attract insects as a collaboration. I'll use colour to attract you. And I use colour based on the original plant, and I use it to bring out kind of structural characteristics of the specimen, and I use it to bring out the character of that specimen. So, whether it's very sexual or whether it's very sculptural. I realise that scientists also manipulate their material constantly because they need to prepare it to show what it is that they want to be seen. And so I became much more confident in my ground, really. I'm also interested in the way that plants will use dispersal mechanisms for dispersing seeds or pollen. I mean, I use dispersal mechanisms through publication, through the internet. So these are a mixture of seeds and pollen. The great thing about SEM is that you can get phenomenal resolution. This is actually technically it's fruit from a small scabious, creeping scabious. But it's, and typically the botanists would show it normally the other way up. And the first time I did one, I showed it that way up. And now when I saw that I did another one, I thought actually, I turned it around. So it's very kind of balletic, very intense. I mean, when it's printed up kind of large, the colour is very, very intense. This is a specimen from one of John Ruskin's collection of plants from Chamonix. So he went over the Alps, like Goethe, collecting plant material. And I drive over the Alps every year to Greece. And I collect material in the filter of the engine of the car. So this is the kind of lazy botanist's guide to collecting. And these are images which are just about to go into a show in Plymouth Peninsula Arts about bees, apiculture. It opens in 2 or 3 weeks' time. So these, some of them are quite easy to identify. They go in kind of families, others more vaguely. Then I work with other people. So this was a project I did for Macallan's Whiskey Distillery. And they were developing a new business centre and they wanted it different. They didn't want the Haggis Trail, so they wanted to kind of bring out the science behind their production. There's not much in the production, it's just barley, water, yeast and starch. So it's a kind of challenge to bring that out. So this is the barley grain cut in half. And as you zoom in, you can see the kind of cell wall. And if you go in further, you can start to see the kind of starch, which in turn will develop to sugar, which will develop the alcohol for the whiskey. But in looking at that, I'm also kind of looking outside. So when I'm looking everywhere, I start to make links between different things. So this is it's a rock structure in Limnos in Greece. Similar forms, very different kind of form of construction or evolution. So, in terms of dispersal mechanisms, this is an ant, again outside my studio. So the ant will take the fruit and seed, mainly medicargo, and take them down to the nest, they'll consume the seed, and then reject the husk. And I work with those. So this is again another medicargo seed prepared. It's splutter coated with platinum. And this is how I develop the images. And here's a kind of reconstructive process. I'm pushing the scale, the possible scale that you can get within the SEM. It's very much at the limit. By doing that, I can create images which I can blur up to this kind of size without losing any kind of resolution. And then I can examine different specimens within the same family. There are 43. I've got a long way to go. I've only got up to 4, I think, for the moment. And that's the only one, that's a tree version, arboreal neck. And I also work in different ways. So these are Naples garlic. And this is my kind of studio workshop at the Gulbenkian Science Institute in Portugal, where I worked for 5 months in the lab there. And in this case, instead of adding colour at the end, I'm adding colour at the beginning. So the blue dye, I told you like blue dye, and I'm cutting up sections from wild plant stems, which I am staining and then photographing. And the stain is bringing the colour to the specimen. I'm not adding colour in any other way, manipulating it. So it shows that the epidermal cells kind of often come out pinkish, chloroplast. And then you can see the whole image. And these are often made up of about up to 600 separate images, which I can deposit and put together. So I can blow them up to this size. There's a whole collection of these, and they're all stained with the same stain. But the proteins in the tissue respond differently. So marigold, primula, that's actually stained with supplements. It's another orchid. And I'm doing time.
That's fine.
And then other contexts. So I just sort of finish off by showing some images from a show I have on at the moment. This is a place called 40 Hall, which is in Enfield. It's an unlikely place if you know Enfield. It's something of a surprise when you turn off the A10. It's a very beautiful house and recently very beautifully restored and run by Enfield Council. And it was built in 1635. So they asked me if I would do a show there. So when I started to research, I found that 1635 was an interesting year because the person that made this, Anthony van Leeuwenhoek, was also born in that year. So this is one of the really kind of first operating microscopes. What was interesting was that Raymond, who built the house at 40 Hall, was a draper. And I was aware of Leeuwenhoek, but I hadn't realised that he was also a draper. And he was developing this so that he could also look at kind of fabrics, as well as looking at plants. So that gave me an in for the show. And I used some of the plant section images printed onto silk, which were hung throughout the space. This is through a stained leaf of sphagnum moss. And also, in the parlour, this dining table. And on the table, there's a tablecloth, and it's printed with a microscopic image of itself, in strands. And on top of which are images of pollen collected from plants around the house. And the plates have a print of section through one of the branches of the mulberry tree outside. And the green plates are also mulberry leaf profile with a magnified image of its stomatal cells and hairs on the back. We're using this as the venue for a filming for the Lens on Life project with artists and scientists coming together quite soon. So that's a pollen grain from the mulberry. And as you walk through the house, I've taken sections and hung on the original rails that the kind of tapestries would have hung from. Fragments of the cell structures. The house is very plain. There's a little bit of furniture, which is, means it's kind of it's much more open for the spectator to kind of occupy. Again, it's another version of sphagnum moss, using kind of polarizing filter. And there are kind of clues around the house as well. Some glass pieces on the shelf with oak moss and some fresh moss, which we kept growing from the site up there. And I also like to be able to respond to things there. And so this room has some other things in which made it quite difficult to show. So I thought the fireplace was a kind of quite a strong pull. I thought it would be quite nice to hang from the fireplace. So it's now tied up, and when someone opens the front door downstairs, the draft through just pulls it up, and when the door closes, it just sinks down again. So it has a life. So there's a kind of passage through the house and through my different ways of working. There are other objects, there are galleries with drawings. At the top of the house, there's a table, 2 sheets of glass really, with flowers that I collected from the Lee Valley nearby and pressed. And the drawings in that space. So it's a kind of specimen table. Some of the people that I've worked with, scientists are always very good at acknowledging that because they work in teams. Artists don't often acknowledge, although they're getting better. So these are the people that I work with.